We pick up part two of our respiratory lecture. We're going to start where we left off, which was talking about gases and how they interact with each other and how they interact with liquid. And then we'll look at some of these gases and pressures of these gases throughout the body. So just in general, if we look at a couple laws, we just covered Boyle's law that said that there's an inverse relationship between pressure and volume. We have two more laws to look at, and this is going to deal with the exchange of oxygen and carbon dioxide. The first law is Dalton's law, and basically this says that each gas, whether it be oxygen, carbon dioxide, or any other gas, in a mixture of gases, exerts its own pressure as if no other gases were present. So what this essentially means is that oxygen is going to have its own partial pressure and carbon dioxide is going to have its own partial pressure, regardless of oxygen and carbon dioxide being together in the mix. Henry's law is going to state that the quantity of a gas that will dissolve in a liquid is going to be proportional to the partial pressure of the gas and its solubility coefficient, assuming that temperature is going to remain exactly the same or remain constant. Now, we are going to look at what happens to the amount of gas that's dissolved when temperature changes. We'll look at that when we look at the oxygen saturation curve a little later in this lecture. But again, Henry's law is that the amount of gas that's going to dissolve in a liquid is going to be proportional to the partial pressure. So again, we see that pressure is going to be directly related to the amount of gas that's going to dissolve in a liquid. The next thing we're going to talk about is just a general idea of the exchange of oxygen and carbon dioxide in the body. So we go back to our different types of respiration. Our external respiration, if you remember, is on a lung level, right, pulmonary level. And then we go to internal respiration, which is on a systemic or tissue level. So pretty much we talk about loading and unloading, loading oxygen, unloading CO2, unloading oxygen, and loading CO2. So during external respiration, so again, if you think about this at the level of a lung, a pulmonary level, oxygen will diffuse from the alveoli into the pulmonary capillaries, and CO2 is going to move in the opposite direction. So quite simply, we're loading up oxygen and unloading CO2. During internal respiration, we see the opposite. So oxygen is going to diffuse from the systemic capillaries into the tissue, and CO2 is going to move in the opposite direction. So again, to make this a little more simple, we see that we're going to have oxygen unloading into the tissue and CO2 loading back in the blood. So whether we're looking at the pulmonary level or systemic level, they are opposite, right? So again, to recap, on a pulmonary level, we are loading oxygen into the blood, unloading CO2 out. And on a tissue level, we are unloading oxygen into the tissue and loading CO2 back into the blood for removal. So we can put this to a diagram so we can look at both circuits, what's happening at a pulmonary level, a tissue level, look at the partial pressures all together, and then we're going to put this to graph. So we can start right here with our deoxygenated blood, and we see that the partial pressure of oxygen is about 40 millimeters of mercury. Our partial pressure of CO2 is about 45 millimeters of mercury. We're going to go to the top, and we're going to fill in what's kind of missing here. Atmospheric air, the partial pressure of oxygen is going to be about 159, and the partial pressure of CO2 is going to be about 0.3 millimeters of mercury. Now, when we talk about what's happening on a pulmonary level, we see our arrows here, and we're going to have to draw them in and fill them in. So CO2 is going to be exhaled, and O2 is going to be inhaled. This is again called our pulmonary ventilation or breathing. We see that the pressures are going to be a little bit different. The partial pressure of oxygen is going to be 105 millimeters of mercury, and the partial pressure of CO2, carbon dioxide, is going to be 40 millimeters of mercury. Now this is going to be important. Remember this number, this 105 millimeters of mercury at the pulmonary, at the lung level, because this is specific pressure. This is the pressure that's going to be required to bind oxygen to our hemoglobin. Again, when we do our oxygen saturation curve, we will explain this in a little more depth. Now we look at our oxygenated blood, and we're moving to our tissue level. We see that our partial pressure is not really changing much. partial pressure of oxygen is about 100, and our partial pressure of CO2 is 40. Now the big change is going to be down here at the tissue level. So let's star this. Now why is this going to be a big change? Because now we have to kind of undo what we did back up here at the pulmonary level. So in order to release some of this oxygen and pick up the carbon dioxide into the blood and release that oxygen into the tissues, we need a pressure change. And we see that. We see right here at the tissue level, the partial pressure of oxygen is 40 millimeters of mercury. And this is going to be important. Our partial pressure of CO2 is relatively constant, a little bit higher at 45 millimeters of mercury. Okay. So we can take what we just learned on our last slide and now look at the logistics of oxygen and carbon dioxide in the blood and what exactly is happening when we say we're loading and unloading oxygen and carbon dioxide respectively on each part of our body on a pulmonary and systemic level. So we can start with oxygen and look at the two ways that oxygen is going to be carried in the blood. So we start with 98.5% of that oxygen is going to be carried by hemoglobin, and we're going to call this oxyhemoglobin. Okay, and we can measure this. If you put a pulse oximeter on a patient's finger and you get a reading of about 98 to 99% saturation, this is pretty normal. This is what we expect because 1.5% of that oxygen is going to stay dissolved. It's going to stay dissolved inside that plasma. So again, 98.5% is a pretty good number. We are not binding 100% of oxygen. 98.5 is binding to those hemoglobins in our red blood cells, and 1.5% of it is going to stay dissolved in plasma. Now, carbon dioxide is a little bit different. We get three scenarios for this, three different ways that we're going to transport it. 70% of the carbon dioxide is going to be transported as bicarbonate ions. And we're going to look at this conversion in a little bit. It's not yet bicarbonate ion. There is a conversion that has to occur. So from CO2, we go to carbonic acid. And then from carbonic acid, we're going to convert to bicarbonate. And we're going to see shifting of hydrogen ions. And this is going to be important when we talk about acidity in blood or basicity in blood. So keep that in the back of your mind when we look at red blood cell further on in this lecture. 23% of the carbon dioxide is going to be carried by hemoglobin inside the red blood cell. And similarly to how oxygen is carried by the hemoglobin, we call it oxyhemoglobin. This carbon dioxide is going to be called carbaminohemoglobin. So we see that word right here, carbaminohemoglobin. So 23%. 7% of carbon dioxide is going to be dissolved in plasma, similarly to oxygen, but a much larger percentage of that. Now we get a nice little slide that's going to give us a diagrammatic representation of what we just talked about. So if we look at the top and we see our transport of carbon dioxide, again, we see 7% is dissolved in plasma, 23% as carbaminohemoglobin, and 70% as bicarbonate. Okay. Now, we deal with transport of oxygen, again, exactly what we just said. 1.5% is going to be dissolved in blood, and then 98.5% is going to be dissolved or inside that red blood cell bound to hemoglobin as oxyhemoglobin. We go to the systemic level and we see 1.5% that's dissolved moves out into the tissue, and then we get for carbon dioxide 7% again back being dissolved in plasma, 23% bound as carbaminohemoglobin, and 70% carried through the blood as bicarbonate. So we're going to shift a little bit. We're going to talk about the factors that affect the affinity for hemoglobin, of hemoglobin for oxygen. And we're going to take the next 5 slides and we're going to kind of merge them into one tutorial that will all this together and look at all the factors that affect the affinity. How much, so if we define affinity, how much that hemoglobin and oxygen like each other. Okay, so again, we're going to pause here. We're going to continue with this lecture at this slide in a few minutes, but let's go to a separate whiteboard tutorial on the oxygen dissociation or saturation curve. Welcome to the second part of our respiratory lecture. We're going to pick up where we left off, which is looking at oxygen and carbon dioxide exchange at a pulmonary level and at a systemic level at our capillaries. So we're going to start right up here on top. So we look at, and just select our pencil. So we're going to start right here on top. So let's look at what's going on. We can start with oxygen. Oxygen is being inhaled, so we inhale oxygen. It moves through the capillary wall and then dissolves and moves into our red blood cell. Now, once inside the red blood cell, we have this combination of hemoglobin and hydrogen. Now, hydrogen is bound to hemoglobin at this point as a result of our internal respiration system that we'll look at in a little bit, and that hemoglobin hydrogen combination kind of reduces the affinity of oxygen for hemoglobin, but here we're going to reverse it. So we're going to pop off that hydrogen and make our oxyhemoglobin. Once that hydrogen is free within that red blood cell, it is going to associate with bicarbonate to make a weak acid, carbonic acid. Once carbonic acid is available, we're going to use our catalytic enzyme, carbonic anhydrase, which is going to catalyze this reaction and separate carbon dioxide and water. Now, remember that we had some carbon dioxide bound to hemoglobin. We will separate it up here as well, and this will give us carbon dioxide to diffuse out of the cell and then respire out. One last thing to look at, I'm going to change the color to green so that we can circle this chloride shift. So we have to understand that right at this point it is a reverse chloride shift, meaning that chloride is moving out of the red blood cell. And then we have to say why. Why is this occurring? Why are we shifting chlorine, a negative ion, out of the cell? Well, this is giving us a electrical balance. We have to maintain a pretty stable electrical conductivity within this red blood cell. So what happens is we have all this bicarbonate, which is a negative buffer that is moving into the cell. So the balance is leaning more negative. So as a result, to equal out that balance, we get chloride right here shifting out, equaling, allowing that balance to remain relatively constant. Now, if we look at a systemic capillary wall, we're down here now. Let's go back to black. So we're down here now, looking at this lower picture, and let's see what's happening. So we have an idea of what's exchanging. So exchange of oxygen and CO2. This is systemic capillaries. This is internal respiration. So what we want to do is unload oxygen and load up CO2, and that's exactly what happens. So let's take a look at what's going on. Here comes CO2. We need to load it. It's going to diffuse through that capillary wall. It's going to move its way into the red blood cell. It's going to use carbonic anhydrase. So again, we're catalyzing this reaction to make carbonic acid. Again, this is a weak acid, so hydrogen is going to dissociate relatively quickly. So as hydrogen pops off, it is going to bind to hemoglobin, which in turn will lower the affinity for oxygen. As we get to our next step, oxygen pops off, dissociates out, dissolves out, and will dissolve to the stem capillary and oxygenate our tissues. Now, here's our scenario. What happens to that bicarbonate once we get rid of that weak acid, pop off the hydrogen? Here comes bicarbonate diffusing through the red blood cell again. Now, the inside of this cell is lacking a negative balance. So what happens is, so that the inside of this red blood cell does not become too positive, we now resort back to, let's go to green again, resort back to our chloride shift. We bring that negative ion in, and it balances the negativity inside that red blood cell. The next portion of our respiratory system that we're going to talk about is control. How do we control breathing, whether it be inspiration, expiration, eupnea, quiet breathing, forced inspiration, forced expiration? We're going to look at those various parts. We're going to divvy it up into two basic regions. We have our respiratory center. Our respiratory center is going to be our pontine nuclei, so we have a pontine respiratory group. From there, we deal with the medulla oblongata. The medullary respiratory center is going to have three basic portions. A dorsal ventral group. That dorsal ventral group, we follow those neurons, are going to connect down to the diaphragm, as well as our external intercostal muscles. Then we have our ventral respiratory group. Now again, notice before you go any further, notice the terminology. Ventral meaning in front, dorsal meaning in back. And if you look at its orientation right here, one is clearly in front and the other is clearly in back. Now, the ventral respiratory group has a secondary complex called the pre-Botzinger complex. This complex is a cluster of nuclei that's going to govern and regulate just normal respiratory rhythm. So the quiet breathing, the eupnea that you're doing now is going to be governed and regulated right here in this pre-Botzinger complex. Our next step in this process is to examine in a little more depth the dorsal respiratory group and the ventral respiratory group, and exactly what happens when these neurons are activated and inactivated. So we're going to start right here. We'll start number one with the DRG. When activated, it takes approximately 2 seconds. We have two muscle groups that are going to be contracting, the diaphragm and the external intercostal. So these muscles contract, and this is going to be their most active phase. Is going to give us our normal quiet inhalation. So again, we're talking about tidal volume. We're talking about these small amounts of air, 300 to 500 mils going in and out during eupnea. When the DRG is inactivated, our two muscle groups again, the diaphragm and the external intercostals, are going to relax and become less active. This is also going to work in conjunction with the relaxation of the elastic recoil of the lungs, which gives us normal quiet exhalation. Notice we are specifying quiet. So when we talk about the dorsal respiratory group, we're talking about the eupnea, the quiet breathing that occurs, the breaths in and the breaths out. The muscle groups we're using are our primary respiratory muscles, the external intercostals, the diaphragm, etc. As we move on to forceful breathing, so forcefully inhaling or forcefully exhaling, we look at the dorsal respiratory group, so it can work in two pathways, one on its own or two by activating the VRG. So on its own, the DRG is going to contract those two muscle groups again, the diaphragm and the external intercostals, which will give us at its most active stage a forceful inhalation. Step 2. Our ventral respiratory group will be again activated by the DRG. This is going to increase the activation of these forceful inhalation neurons. We now innervate those accessory muscles of inhalation, those secondary respiratory muscles, the sternocleidomastoid, the pectoralis minor, the scalenes. These three muscle groups are going to contract to give us a big forceful inhalation. Now that deals with exhalation. So that covered inhalation, now exhalation. Again, the DRG activates the VRG, and we get activation of our exhalation respiratory muscles, our internal intercostals, our external oblique, our transverse abdominis, our rectus abdominis, and our internal oblique muscles will all contract and will give us a forceful exhalation at their most active stage. So that is going to deal with our control of respiration. We're going to have one more slide that's going to give us a little more idea or peek into the window of what controls respiration, and that deals with cortical influences. These cortical influences are going to allow conscious control of respiration that are going to be needed, especially when we have to avoid inhaling noxious gases. We also rely on chemoreceptors. These chemoreceptors are going to be located centrally and peripherally, and their job is going to be to monitor levels of oxygen and levels of carbon dioxide, and it's going to provide input to the respiratory centers. So the question now is, where are chemoreceptors found in these cortical reflexes? So we move on to the actual location of these receptors. So we see number one, on the arch of the aorta, we see aortic bodies. Okay, these are going to be detecting the chemical changes in blood as it leaves the heart in the aorta. Now that makes sense. We're looking at oxygen level concentrations, CO2 level concentrations, and as a result, if, say, oxygen levels deplete or CO2 levels increase or vice versa, these chemoreceptors will detect it and send feedback back up to the brain, back up to the medulla oblongata, back up to the pons, those pontine nuclei, and tell the body to change its respiratory pattern. The second place we see it are within these carotid sinuses. So again, look at these important places. We're going to call them our carotid bodies. Now, why? We're monitoring blood before it actually gets to the brain. So that's super smart too, right? So look at these two major important places. The blood that's leaving the heart that's going to all over the body, and the blood that's going to the brain. Remember how much oxygen and nutrients the brain actually needed. So we need to ensure that we maintain a high level of oxygen. If oxygen levels drop, we know that symptoms from the brain can be dizziness, mental confusion, haziness, etc. So our respiratory pattern will adjust once it detects these low levels. We can now name these conditions that we've been talking about where we have oxygen deficiency or too much CO2 detected by these chemoreceptors. So our first condition is going to be called hypercapnia. What this is a slight increase in the partial pressure of carbon dioxide, which ultimately increases those hydrogen ions. And this is going to stimulate those central chemoreceptors. What's going to happen is it's going to increase respiratory rate and increase oxygen. When we have hypoxia, so hypo means low, oxia, oxygen, so we get an oxygen deficiency at the tissue level. This is caused by a low partial pressure of oxygen in the arterial blood. And conditions that can cause this might be high altitude, say you went to a football game or you moved to Colorado by Mile High Stadium. Another condition that can cause chronic hypoxia could be some type of obstruction in the airway or fluid in and around the lungs. We can now take the control of respiration and put it all together. We can look at a feedback loop and say, what happens when we have a disruption? So above this, I'm not sure why it got cut out, is a circle that should say stimulus. So we get a stimulus that's going to disrupt homeostasis by increasing a controlled condition. The controlled condition is going to be the arterial blood. So what we're doing is we're taking the partial pressure of CO2 by decreasing pH or the partial pressure of oxygen. What happens is the central chemoreceptors in the medulla detect this. We deal with our carotid bodies, and our aortic bodies are also going to detect this decrease of oxygen. And what's going to happen is we're going to send impulses. These nerve impulses are going to go to the dorsal respiratory group in the medulla oblongata. As a result, we're going to send nerve impulses to our effectors, the muscles. These muscles of inhalation and exhalation are going to contract more forcibly and more frequently, giving us a state of hyperventilation. Our end goal is to increase oxygen by decreasing carbon dioxide concentrations in blood. So here's our response. So our response, decrease arterial blood PCO2 of carbon dioxide, increase in pH, and increase in the concentrations of oxygen. As this happens, we will return back to homeostasis when response brings that arterial blood CO2, pH, and O2 back into normal range, and this will negate our system. Now, we can look at other types of scenarios with somebody who has or is hyperventilating due to stress, and the receptors are detecting high levels of O2, so we're hypersaturating our blood with oxygen. How do we respond to this? Well, it's a pretty simple solution. We take a brown paper bag, we make a modified rebreather. What we're doing is we're forcing that patient to rebreathe the CO2 that they're exhaling, which will temporarily and artificially remove the available oxygen, therefore lowering the oxygen content in the blood. These chemoreceptors will detect that. It will shut down this hyperventilation system, or these nerve impulses that are causing the hyperventilation, which in turn will allow and bring us back to normal breathing. Table 23.3 is a good table that's going to help us summarize the stimuli that affect breathing rates and depth. So we can look at some of these examples. So stimuli that increase breathing rate and depth, we look at voluntary hyperventilation, which is controlled by the cerebral cortex, and anticipation of activity by stimulation of the limbic system. What happens? We get voluntary hypoventilation controlled by the cerebral cortex. This is a stimuli that's going to decrease breathing rate and depth. So some more stimuli that increase breathing rate and depth. We increase in arterial blood, CO2, above 40 millimeters of mercury causes an increase in hydrogen ions, which will be detected by peripheral and central chemoreceptors. On the flip side, stimuli that decrease breathing rate when it's related to the partial pressure CO2, so decrease in arterial blood, PCO2 below 40 millimeters of mercury causes a decrease in hydrogen ions. This is going to be detected by the peripheral and central chemoreceptors and will adjust by decreasing breath or breathing rate. We can increase the activity of proprioceptors. We could also decrease the activity of proprioceptors, again going to increase or decrease breathing rate accordingly. When we're dealing with exercise in the respiratory system, and again, I apologize that your slide's going to look a little bit different than mine. The exercise and the has been deleted for some reason when we migrated this slide over. But again, the slide is exercise in the respiratory system. We notice that the respiratory and cardiovascular systems work together. They work together to make adjustments in response to both the intensity and duration of exercise, right? So you can think about this. Think about when you're running on a treadmill and you just start out, your breathing's relatively normal. As your body starts detecting, those chemoreceptors start detecting increase in CO2, your respiratory rate will start responding by increasing to account for that oxygen indebtedness. And in response, your cardiovascular system is also going to increase. Your heart rate will increase to meet the demand of the increased demand of oxygen. So as cardiac output rises, the blood flow to the lungs, termed pulmonary perfusion, so we see that word here, pulmonary perfusion, will increase as well. The oxygen diffusing capacity will increase threefold during maximal exercise, so there's a greater surface area available for oxygen diffusion. The next slide we look at is aging. So when we discuss aging in the respiratory system, we have to talk about what happens as we get older, and this seems to be a common thread with all our systems. As we get older, our respiratory system decreases in its functionality, its ability to function properly. So what happens? As we get older, compliance decreases. The ease at which we're able to breathe and expand the thoracic cavity and those alveoli, the elastic recoil within those alveoli decreases, which means that our ability to breathe forcibly in and forcibly exhale, so forcible inhalation and forcible exhalation decreases, which means that overall, number one, our vital capacity will decrease. Our blood oxygen levels tend to decrease as we get older, again, due to long-term damage and lack of compliance from our respiratory system. Our alveolar macrophage activity, so this is 2. Number 3, our alveolar macrophages stop working as optimally as they did when we were younger, which means that this will make us more susceptible to dust, dirt, bacteria, debris that actually makes its way down into those delicate alveoli. So those alveolar macrophages usually take care of this when we're young. As we're older, they start decreasing their activity. Lastly, ciliary action, number 4. So those cilia stop working, they're damaged, they're not functioning properly. So that first level of protection that we talked about way back at the beginning of this lecture has been removed or reduced. So we lose that protection in that respiratory epithelium that is allowing us to filter, trap that dirt, that debris, those bacteria and those viruses. So ultimately, consequently, elderly people are going to be more susceptible to things like pneumonia, to bronchitis, emphysema, and other issues. Brings us to our last slide of disorders. So we look at different types of respiratory disorders that disrupt homeostatic balances and brings us into homeostatic imbalances. The first being asthma. So we should be pretty familiar with that. This is a condition that causes inflammation of the bronchi, that bronchial tree, that makes breathing very difficult. Labored, we have chronic obstructive pulmonary disease, or COP, again, something that is obstructing the pulmonary pathway. Lung cancer, pneumonias, tuberculosis is another bacterial infection that affects breathing. Common colds, increase in mucus productions, fluid buildup in and around the lungs, pulmonary edema, cystic fibrosis, so a hardening of the lung tissue, which makes compliance really, really hard. Mesothelioma, which is related to asbestos exposure. And then sudden infant death syndrome and acute respiratory distress caused by viruses like RSV, etc. So this is going to conclude our lecture, and we end with the focus on homeostasis and what our respiratory system has to do with maintaining balance, keeping us alive, working with our endocrine system, our cardiovascular system, our nervous system, and our muscular system to give us a happy, healthy respiratory function.